All right, well, welcome everyone to the Barley Pest Initiative Virtual Forum, February of 2025, reporting on the year of most of the work being done in the year of 2024. Thanks everyone for joining us today. I do believe we've got a few newer or new-ish faces, and so I do think it would be helpful to just go around and just do some real brief introductions. Just go ahead and say your name. And let's actually just say what the primary pest is that you work on. I know many of you work on several. Just pick either your primary or maybe your favorite. So with that, I will just call names out so that we can kind of go around in a rapid fashion. And I will start with Tanner.
Hi, so my name is Tanner Shan. I'm the lead scientist for the Grain Genes project. I guess the pest I'm working on is computer viruses.
Good answer. Good answer. Gary Bergstrom.
Hi, everybody. Gary Bergstrom, Barley Scald, Rhynchosporium.
Nice. Matthew Moscow.
Hey everyone, I work mostly wheat stem rust, but I'm also going to say barley stripe rust since I care about them both deeply.
That's good. Good. Doug.
Yeah, Doug Luster, USDA ARS Fort Detrick, and I work on all the cereal rusts and about 10 other pathogens.
Roger. Roger, you're on mute.
Yeah, as a meeting.
Apologize for that.
How can
Roger
this
Wise,
live?
I'm an ARS scientist in Ames, Iowa. I work on powdery mildew.
Thank you. Thomas.
Thomas Baldwin, North Dakota State University. I work on bacterial leaf streak.
Isabel.
I am at UC Davis, mainly breeder, malting barley breeder, but main challenge here are barley yellow dwarf, cereal yellow dwarf, and stripe rust.
Great. Bob.
Bob Brugman at Washington State University, and the main disease is stem rust.
Christina.
Hi, Christina Cowger, USDA ARS in Raleigh, North Carolina, and barley yellow dwarf virus would be the main one.
Timothy.
Tim Paul at USDA ARS in Pullman, and I work on soil-borne pathogens, both fungal and nematode, including Pythium rhizoctonia and root lesion nematode.
Dr. Chen. And you're on mute too. Oh, you're still on mute.
You're talking for the.
Hello, everyone. Now you can hear me. Xianming Chen, USDA ARS at Pullman, Washington. We work on barley stripe rust.
Thank you. Dr. Tatnini.
I'm Satya Tatnani, USDA ARS Lincoln. We work mainly on barley yellow dwarf virus and the cereal yellow dwarf viruses.
Thank you. Belay.
Hi, my name is Balai Yemar, USDA ARS in Aberdeen, Idaho. I work with bacterial leaf streak and fusarium head blight.
Awesome. Dang Ying. You're on mute, Dang Ying. You guys are being so good about mute.
Yeah, this is Dong Ying from USDA ARS in Aberdeen, Idaho. We work on the barley strip rust.
Judith.
Hi, I'm Judy Coltman. I work with Gary Bergstrom and Mark Searles at Cornell University on scald and barley.
Rajiv.
Hey, good afternoon, everybody. This is Rajiv Gupta. I'm the research leader for cereal crops in Fargo. So the unit works on different diseases and pests in barley and some abiotic stresses as well. Thank you.
Melissa.
I'm Melissa Carter. I work at the USDA ARS, and I'm in Dr. Doug Luster's lab here at Fort Detrick, and we work on cereal rusts.
Thank you. Stephen.
Yes, Stephen Wegulo. My video won't start, so I hope you can hear me.
Yes, we can.
Stephen Wegulo, I'm a small grains pathologist at University of Nebraska, Lincoln. And I work mostly on fusarium head blight, but recently we started a project on bacterial streak as well.
Perfect. Mitch.
Hi, I'm Mitch Elmore. I'm a scientist at the Cereal Disease Lab in St. Paul, Minnesota. We primarily work on Fusarium head blight of barley.
Haley.
Hi, sorry, working from home today, but I'm a USDA ARS research scientist or research entomologist, and I mainly work with pests in wheat, barley, and sorghum.
Thank you. Greg.
Yeah, hi, I am Greg First, USDA ARS, and I work in Roger Wise's lab on powdery mildew, barley.
Thank you. Oh, Dr. Zhang just turned your camera on.
Oh. Okay, yeah, thank you. So I'm Xiaofei Zhang from UC Davis. Actually, I joined UC Davis last March, a little bit around a year. So I'm in charge of the small grain crop breeding, including barley. We work on mainly focus on malting barley with Alicia. So for disease, we work on strip rust disease and barley yellow dwarf virus disease. Yeah, thank you.
Thank you and welcome. Kevin.
Kevin Smith at the University of Minnesota. Work on everybody's favorite Fusarium head blight and a few other diseases as well.
Thanks. Dr. Lee.
Hi, this is Ginjia Lee. I'm working with USDA AIS, Stillwater, Oklahoma unit. My research focuses on disease and aphids resistance. Thank you.
Thank you. Lovepreet.
Hello, my name is Lovepreet Singh, and I'm working on Fusarium head blight in Dr. Mitchell Moore's lab at USDA Cereal Disease Laboratory, Minnesota.
Thank you. Dr. Barman?
Hi everyone, my name is Mrityunjay and I'm working in University of Nebraska. Basically, we are looking for barley yellow dwarf virus and cereal yellow dwarf virus strain composition in Nebraska cereal frills. Thank you.
Wonderful. Ransom.
Hey, my name is Ransom Sitz. I work at Anheuser-Busch, and we just have a breeding program, so we don't have a specific pest that we're working on.
Thanks for joining us, though. Now, names have scrambled a lot since we got started, so I'm going to miss some people, but I think Matthew Brook, you might be up.
Yeah, I'm a postdoc at Oregon State University, and I'm working on barley yellow dwarf.
Welcome, Scott Fisk.
Hi there, I'm also from Oregon State University, in addition to, BYDV. Here we're also working on stripe rust and scald.
Well, and we will round out the OSU barley world, Laura.
Hi, as Scott said, all the same. Cypress, scald, and yellow dwarf. Also involved in Fusarium head blight too with DH production. Yeah.
Thank you. Eugen? Dr. Jen, if you're talking, we cannot hear you. Or, go ahead.
Okay. My name is Eugene. I'm working at the Cereal Disease Lab, St. Paul, Minnesota, ARS, working on stem rust of wheat, barley, and oats.
Awesome. Gina.
Hi, I'm Gina Brown-Gadira and I'm working with the ARS in the Eastern Regional Small Grains Genotyping Lab. So we do some genotyping in barley.
Great. Raphael?
I just started my PhD under Dr. Catherine Frous at UNL, and I'm here to observe and learn.
Awesome. Thanks for joining us. Tim?
Yeah, I'm Tim Friesen, USDA in Fargo, and I work on netform net blotch, spot for net blotch, and we're just starting to work on some viral issues in barley.
Awesome. Poria?
Puria and Safi from University of Idaho. I work on Cereus nematode, barley and wheat.
Great. And then Julie. Julie Stevens, if you're talking, at least I'm not hearing you. All right, I think we're going to keep going just for sake of time. If you want to come off mute, you're welcome to do so. But yeah, great. This is actually a wonderful turnout. 37 of us. That is wonderful. Thank you so much for taking the time today. I'm going to go forward to my first slide because I am going to use Slido again this year. It's kind of fun just to actually see these results evolve year to year. And I think it's just a more interactive way for us to engage in this forum, especially since there are so many of us. Might be a little intimidating sometimes to speak. So feel free to go ahead and scan that with your smartphone, or if you just want to open a web browser, you can go to slido.com and type in that number. And we'll have just a few polls coming through. And also, I just want to say again, for those of you that might have missed it at the beginning, I'm kind of on my own today running the webinar. Scott's actually in Washington, D.C. today doing some lobbying work to try to get us more money. And so if you're dropping things in the chat, there's a good chance I'm going to miss it. So please, if it's anything urgent, just jump into the conversation. For those of you new to this forum, this is very informal. This is really just about bringing everyone together and sharing our work, and then also just thinking ahead to the future. So feel free to jump in if you have anything to contribute. All right, so just to kind of go ahead and get this kicked off, we will just start with just to make sure everybody kind of feels comfortable using Slido. Just curious what your affiliation is. So are you a BPI funded researcher, a researcher maybe not funded through the BPI yet, administrator? Do you work with the NBIC or with AMBA? So feel free to go ahead and respond. And then also through Slido, you'll notice that there is a way to drop questions in. And honestly, it might even be a better way to get a hold of me during the webinar because I will see those before probably the chat because I've got the phone open next to me. So feel free to drop questions in there if you think of them. All right, wait for just a couple more responses. No surprise, overwhelming majority of you are all funded through the BPI, which is great, but good to also see some other folks here too. So thanks for joining us. All right, we will keep on going. So just as a kind of a high-level overview, because we do have some new faces here, either people that have just joined research teams or perhaps do not receive funding through the BPI, but just interested about the work. I just kind of wanted to provide the foundation for what the BPI really was established to do. So in the BPI, we are seeking to develop genetic resistance and management strategies to mitigate the negative economic impact of 20+ pests. So we're looking at over 20 insects and diseases in the BPI, essentially everything but Fusarium head blight. And in there, we're targeting insect pests, bacterial, viral, and fungal diseases. So that is what's all included in the BPI. And we're doing that work across over a dozen states, which is really exciting. And it's a wonderful partnership between ARS and our universities. So I took all of the data that you guys submitted to me through the BPI questionnaire this past year and just did some crunching of numbers again. This past year, we actually had over 60 researchers involved in this work, and we represented over 15 states, which is excellent. So we're kind of expanding our reach. Those are the ARS facilities and universities that have been involved. And excitingly, this year, we actually had 12 new publications come from this work. I believe last year we had 9 new pubs, so we've actually increased that number. But that's a really great output showing that there's a lot of great work that's happening across this research. And I really do appreciate everyone submitting that to us because it does provide us a really nice metric on the output of this work. So congratulations to those of you that were able to publish based on some of the research funded through BPI this year. I did want to give you a quick update on our BPI funding progress. It is obviously, and many of you are living this firsthand, a pretty odd and chaotic year right now with the federal government, especially relative to funding. And so the chart here on this slide just shows the trajectory of funding over time. It was established in FY21 with 1 million. We were able to get nice incremental funding increases each year post that, but we have essentially plateaued going into FY25, and we have tentative still written there because we actually have not received an indication of what our FY25 funding levels will be yet. So we're still operating essentially at that FY24 level. Of course, we were supposed to have a new budget back in October 1, but that rarely happens these days. And actually the current continuing resolution that we're working on runs through March 14th. So we're going to have more of the same for probably at least another month, but then hopefully we will have a new budget from Congress. The House version of the spending bill does have a slight increase for the BPI in there, but the Senate version does not currently. So it's really unknown what we can expect. But we are still trying to reach that $5.3 million target. And when we go to DC next month through the National Barley Improvement Committee, some of you on this call will be there with us. We do plan to ask for that full $5.3 million. I do think that it's going to be a very challenging funding year and do not expect to see a substantial increase to the BPI. And in fact, I think that if we can all just maintain funding levels, that's going to be a tremendous success this year. So I do just want to share that with you all, because I know many of you are having to make decisions on how you may or may not grow your programs. And I think we need to be really conservative right now that we likely won't receive a significant funding increase in FY25. But as soon as we get that budget, we will already be working on FY26. So we will keep this funding target in place. And we can have a discussion about that at the end of the forum today too, if you guys have any additional questions on that. And I do just want to thank everyone sincerely for filling out the BPI questionnaire after the forum today. The annual report that's shown here will go out to all of the attendees plus many of our stakeholders so that they can see the good work that has been done. Much of that is going to be shared in our forum today, but I did just want to let you know that that is going to be out shortly, and so you guys can all view that and feel free to share it with other stakeholders too that might be interested in this work. I do plan to change up the questionnaire a little bit now that we've gained a little bit of time with this funding and programs are maybe a bit more established. We will probably change up the questionnaire a little bit, probably make it a little less intense now that we have a kind of better understanding of the work that's going on. But nonetheless, I still feel it's very important to do each year just so we have accountability, especially back to Congress. The content that I collect in this is very helpful when I go and have those conversations to try to increase our funding. And with that, I did want to just mention that when you're reporting, just a few things kind of got flagged out to me this year. So first of all, I do ask that you guys submit photos for each of the pest projects that you're working on. And I have received some tremendous photos because of that. Many you'll see in this presentation or in the annual report. We certainly use them when we're reporting to Congress. But I do just want you all to be mindful that if you're sharing a photo, please make sure it's your own. I know that I've gotten some photos submitted to me that have had copyrights from other people or certainly don't appear to be barley even. I've caught a couple wheat photos coming through. So I know it's a little cumbersome to add photos and you maybe haven't always taken a picture, a great picture, but please just upload something. Make sure it's barley, make sure it's your own picture, because I certainly wouldn't want to be using photos that I don't have permission to use. Also, with regards to publications, this year is the first year I kind of noticed this. We had quite a few publications reported that I know that we had come through last year as well. And just so you know, I do actually check each single one. I go through and check the published date and make sure that we didn't report on it last year. So it would really help me out a lot if we make sure that we only give credit to new publications that you've done in the last reporting period. And then finally, because I do end up having so much content come through to all of you on all of your projects, which is excellent. Sometimes though, it can be a little hard to pick out the nuggets, like the really good highlight success stories. So I do plan to incorporate in the next questionnaire an opportunity for you to just provide me a very short snippet, something that you're really excited about, something that you want to highlight, because sometimes that gets lost in the reporting. And even if you have something right now that you want to talk about from the last year or something that you're really excited about in the year to come, feel free to just send me an email as well, because it's much less likely to get lost in the chaos of the questionnaire database if I have an email directly from you. So just want to throw that out there, because those little short stories are what really resonate well when we're meeting with members of Congress. And I'm always really grateful for getting those from everyone. So with that, I want to leave plenty of time today for our lightning presentations. Now, I am going to say that I did ask for 5 slides from our 4 speakers, and I got anywhere from 10 to 17 slides. But I am going to keep each speaker to 10 minutes just because I want to get through all 4 of you. But all jokes aside, I do really appreciate some of you and your willingness to present here. So I'm going to share everyone's slides, except Tanner. I didn't quite get his uploaded today. But we are going to start with Washington State University. So Bob Brueggemann, you want to go ahead and come online, and then I will just progress your slides for you.
So you want me to just tell you when to advance?
Yeah, tell me next, and I'll try to keep up with you.
I have 10 slides.
Are you going to send this out, these slideshows out after the whole thing's over?
You want me to send the slides out?
Yeah.
Yeah, usually what I do is I post them when I post the forum recording and also the annual report. I'll put the slides if I've gotten permission to share them with the group.
All right, I just want to be able to listen. So.
Oh, sure. Yep. Nope, that's fair.
All right, you can, okay, thank you very much. So I just wanted to start out talking about the stem rust work that we're doing here at WSU. The story starts back when I worked here for probably about 15 years on stem rust. I'd never actually seen the disease in the field in Washington State. But when I returned to WSU in 2019 after a 10-year time at North Dakota State University, come to find out that stem rust had become a perennial issue on barley and club wheat over the last decade. And so we took it upon ourselves to go out into the wild and into the fields, and we collected a stem rust Uridinea off of barley and club wheat, as well as Aetium off of the secondary sexual hosts, barberry and Mahonia. And my student Arjun Apadaya, who was working with me at the time, he isolated 200 single pustule isolates from the spores that we collected. And from that, we did virulence testing on all the known barley resistance genes at that time. And alarmingly, we found that this population had isolates that were highly aggressive and virulent on all known stem rust resistance genes in barley, including the stack of RPG1 and the RPG4-5 complex, which hadn't been reported worldwide previous to this. And the most aggressive isolate that became our workhorse isolate for the work that I'm going to be talking about today was this PGT isolate LSP21. Next slide. And so the interesting thing about this story and the major mystery is that almost 100% of the isolates were virulent on RPG1, about 16% were virulent on RPG4-5, and 10% of the population was virulent on the RPG1, RPG4-5 stack. But interestingly, neither of these genes had ever been deployed in any barley varieties in the Pacific Northwest. So there's a major question of why is this virulence evolving against these genes, and where is it evolving? And one of the hypotheses that we have is that it's possibly evolving in the natural ecosystem as stem rust cycles between Mahonia and barberry and the wild grasses. So to be able to start filling pieces in that puzzle and understand this mystery, we put together an association mapping panel of 100 of these diverse isolates and sequenced all of them to about 100x coverage, and identified about 100 or 1.2 million SNPs and ran an association on it, and were able to identify a really nice locus for the AVR RPG1, as well as 2 loci for AVR RPG 5, 4, 5, or the RMRL complex. And so we're moving forward on trying to validate these genes. That works moving slowly, but we'll get there eventually. Next slide. So moving to resistance, we screened the World Barley Core Collection, and we identified a handful of lines that had moderate resistance at the seedling stage, but none of this resistance held up in the field when we went to the adult plant stage. And we're not sure if it's because it's temperature sensitive or not, because we had really high temperatures during those years. So we're continuing to work on checking that out. But we turned to the Wild Barley Diversity Collection that was put together by Brian Stephenson et al. And we identified 2 lines, WBDC 74 and WBDC 191, that had a very high level of resistance to this PGT isolate LSP21. And you can see these 2 on the left-hand side, and then some of the RPG1 and RMRL and susceptible checks on the right. Next slide. So interestingly, in 2017, Brian Stephenson group had showed that these 2 lines also had a high level of seedling resistance against some PGT races, as well as a rye stem rust isolate 92MN90, and they designated this the RPG7 gene. They put together a biparental population of high proly and WBDC 238, and we're able to map the gene to about a 10 megabase pair region on chromosome 3H that contained about 300 genes. So we started collaborating with Brian on trying to do high resolution mapping and positional cloning of RPG7. Next slide. And so we developed a population of Morex by WBDC 94 and screened about 474 F2 individuals for about 948, I think, recombinant gametes. We use these 2 lines because Morex has the genome assembly, and we had the WBDC 94 pangenome assembly, which Brian allowed us to utilize. So we were able to develop a marker panel of about 45 amplicon SNP markers across the RPG7 region, and we're able to genotype those and put together a high resolution map. Next slide. And from the high resolution mapping, we're able to delimit RPG7 to about a 0.5 centimorgan region that was delimited by 5 critical recombinants to a physical region based on the Morex version 3 genome assembly to contain 4 candidate genes. Our top candidate gene is an RPM1-like a nucleotide binding site leucine-rich repeat protein, which, as most of you probably know, is the major resistance type genes. There was 2 F-box proteins and also a glutathione S-transferase in the region. When we did allele analysis between Morex and WBDC 94, there's no polymorphism for the 2 F-box or the glutathione S-transferase. So RPM1-like NLR is our top candidate gene in the region. Next slide. And so to begin to start validating if it is the correct gene or not, or if it's one of the other genes in the region, we did a forward genetic screen. Brian provided us with a lot of seed of both WBDC 94 and 248. We did an EMS mutagenesis on it, and we screened thousands of M1 generation single seed and identified several putative mutants. As you can see, WBDC 94 is on the right with a high level of resistance. It's usually about a 0 fleck. And then you can see some of the mutants that we identified on the right in that WBDC 94 background. And then we also sent 1,000 M2 families to Brian, which he screened and identified some putative mutants independently as well. So he's working on sending us that seed shortly. And so next slide. So we're beginning to sequence the candidate genes across these mutants, and this is data that's fresh off the press. We just recently found that this 94M1-15, which was shown to go from resistance to susceptibility at the M1 and in the M2 families, has a single G to A substitution, nucleic acid substitution in the nucleotide binding site, which results in a serine 238 to proline amino acid substitution. So we think this might be a good mutant within the RPM1 like NLR. And so we're currently doing some genetic analysis to determine if this nucleotide substitution co-segregates with the susceptibility. And hopefully we'll find some additional mutants and also map some of the other mutants that don't look like they're in the region to see if there's something in the pathway or downstream of this recognition. And so with that, next slide. To utilize this, we've developed pace markers for the RPG7 gene, and they are specific to WBDC94 allele. So once we validate it, these will be perfect markers to track RPG7 in the program. We have made many different crosses to integrate this into our breeding program and are currently utilizing this marker to be able to start some variety development that contain the RPG7 gene that will also be stacked with a source of adult plant resistance that came from one of a double haploid population out of OSU's Pat Hayes group, as well as RPG1 and RPG4-5 complex. And I think I might have made it on time.
You were right on 10 minutes. Good job, Bob. Thank you so much. And Doug, if you want to go ahead and get ready, we do have maybe time just for one question, and we'll do some more questions at the end. But does anybody have anything pressing right now they'd like to ask Bob? And you're also more than welcome to drop them in the Slido Q&A as well.
Bob, do you have any idea on the persistence of this gene in the population?
What do you mean, as far as within our population or within the world or the wild barley diversity?
In yours.
In our population, it segregates as a single dominant resistance gene in a perfect, well, not perfect, in a 3-to-1. So it is a single gene. And I know I kind of jumped to the hypothesis that it was RPG7, but we did show that it does map to the RPG7 region as well as with the fine mapping.
Awesome. All right. Well, we're going to go ahead and go to the next speaker. And then when we have some time at the end, we can do additional Q&A of our presenters today. So with that, Doug, if you want to go ahead and take off and just let me know when you want me to progress the slides.
Yes, thanks. Yeah, thanks for the invitation. And thanks for you and BPI for the work you do with our ARS appropriations. So we've already introduced ourselves. I just want to say Melissa Carter in my lab is the one that actually does pretty much all of the hands-on work and containment you're going to see. And then this is a collaboration with the Cereal Disease Lab. Pablo Oliveira just joined as an ARS researcher, and then Eugen and Matt are there as well. Next slide, please. So at Fort Detrick, ARS, we have a very large BSL-3 plus plant pathogen containment greenhouse. Was from the 1950s Army program, and we recently modernized it in 5 phases so that it's fully up to APHIS standards. Next, please. So the important part of this is that in that facility, we, under APHIS permits, are allowed to conduct research and put exotic plant pathogens, pretty much any type of plant pathogen, and arthropod vectors on live host plants. And a lot of what we do is actually with rusts. That building was actually built to contain rust. And we have about 4 or 5 projects on rust other than even the cereal rusts. And so our import permits allow us to bring in a lot of different species, and that's pretty much our niche or our role. We're there because we can help with global surveillance efforts for a number of different pathogens and pathogen groups. Next slide, please. So this project that I'm describing is under what we have termed the Disease Early Warning System, or DWAS. This is a Bill and Melinda Gates funded and FDCO funded project that's run under CIMMYT by CIMMYT. And it's a global cereal rust surveillance project. And we actually just recently, or I guess at the beginning, added wheat blast as well. And so it's basically a global surveillance for any cereal rust. It's focusing on PGT, but we get a number of different rusts in. They ship to us under our permit, goes to the plant inspection station in Beltsville first. And everything is up to snuff with APHIS PPQ requirements. Next, please. So the procedure that Melissa goes through is pretty much standard. But in this case, of course, we're recovering and then inoculating on a small set of differentials, all inside the BSL-3 and actually in double containment. So Melissa recovers Uridine spores from any incoming infected material. We make our decisions on the differentials. Like we basically powwow either by email, and it just depends on where they came from, what lines they were isolated from or collected from. And Melissa plants those differentials out. We treat them with maleic hydrazide to increase the susceptibility, and then spray inoculates and go into our custom built dew chambers overnight. And then as the point where they're beginning to show infection, we have to double or secondary contain them. They go into cellophane bags. All the work that is done with the inoculation and recovery is in a biosafety cabinet. Next, please. And then Melissa does photographic symptom recording just to see the infection severity on the differentials. She then harvests them into gelatin capsules, and we archive in different parts of the year. We archive them into liquid nitrogen, but then under APHIS permit, we're allowed to ethanol kill and ship the rust infected tissue to CDL. Matt Muskow does that work at CDL. Next, please. So yes, everything goes to CDL during their winter containment season. That's starting in December. It's a very compressed schedule for them. They're permitted to work in the Minnesota BSL-3 for a short period of time there. So we move things very, very quickly. And at CDL, they do single spore, in-depth genotyping, and race phenotyping. Next slide, please. And it's been a pretty big effort. And I will again say that the base funding increases that we saw from BPI have really helped. That containment facility is extremely expensive to run. So for 27 countries, we've processed over 1,700 samples, archived 1,200. And so we maintain that archive, and we ship a subset of those to CDL so that we have a safe and secure archive here at Fort Detrick. Next, please. This is a very impactful program. It's a global surveillance network. We're certainly not the only lab. Provides early warning of new genetic variants and monitoring of novel virulences to barley resistance genes. It starts a chain of actions by a large group of researchers downstream on epidemiology, pathogen population genetics, post-resistance, breeding, et cetera. And then the important isolates that show unique virulences are used to test barley breeding germplasm. And a big picture, it is a global effort. We support global food security through breeding for durable resistance. And last slide, I think, is acknowledgements. This is under the umbrella of the BGRI, or Global Rust Initiative. So again, we're partnering here with CIMMYT, FCDO, Bill and Melinda Gates, and a lot of the work is done at the BSL-3 at the University of Minnesota. And so it's supported in part by this grant funds, but then on our base funds, USDA ARS as well. That's my last slide.
Awesome. Thank you so much. And wonderful work on the timing. Are there any questions? Any questions for Doug or other folks at the Fort Detrick site? Roger?
Just like
Yeah.
to say, I think this is awesome.
Yeah, I know it's hearing that. I mean, a lot of you guys are doing very specific work, looking for resistance, but some of these kind of overarching programs that are supporting a wide breadth of labs just is really exciting to hear about this. So I really like the variability in the work that's getting reported today.
Thanks.
All right, great. Well, with that, I'm going to turn it over to Tom Baldwin at North Dakota State University.
Thank you, Ashley. So when I first came to North Dakota, I'm a fungal geneticist. I mostly work on Fusarium head blight. And I asked our serial extension pathologist, Andrew Friskop, what was keeping him up at night, figuring it was Fusarium head blight. And Andrew replied to me that, head blight is a constant pressure, constant pain, but they know how to deal with it and they know how to manage it. What was keeping him up at night was something called bacterial leaf streak, which I've known about through work in Idaho, but didn't really know too much about it. So I said, okay, if we're going to have to know and understand this pathogen, we need to go out, find the isolates, and figure out what's going on with it. So bacterial leaf streak is caused by Xanthomonas translucens, pathovirus translucens. It's a pretty interesting, unique bacterial pathogen causing bacterial leaf streak. If you could flip to the next slide. Thank you. And because just myself, I think, and Belay are familiar with this, I figured I'd post a picture to show you what it looks like in kind of the later stages. It starts out as water soaking of the leaves and then further decays into this chlorosis, really breaking down the chlorophyll of the leaves and causing excess damage. We don't know what the yield impact is on barley. We're going to be working on that. But in wheat, heavy infestation, heavy infection can cause yield losses up to 60%. Which is not good. And because there are no management strategies, we can't use pesticides on this. We don't really know a good management other than crop rotation. We decided to take a look at this and the organism that was causing it. Next slide. So just to give you an introduction to the team, I'm not a bacteriologist myself, but Dr. Zhaohui Liu has been working in this field for longer than I had. He was working primarily on XTU, which is Xanthomonas translucens pathovirus ankylosa, which primarily infects wheat and causes wheat leaf streak. And then BPI is funding 2 graduate students, Jill Velasco and Glenn Rose Belen, who've been working on collecting isolates and running these experiments for the last 4 years. Glenn Rose has been newer to this. Okay, next slide. So what we've been doing is collecting isolates from North Dakota, Montana, and Idaho, which are the barley growing states, as most of us know. Thanks to Juliet Marshall in Idaho and Belay Ymer in Idaho, and Jamie Sherman in Montana for helping us kind of collect those isolates. But we've also been scouring North Dakota pretty well to collect isolates. We now have 120 isolates over multiple years, and we've done full genome hybrid analysis, hybrid sequencing and alignment. And what we find is that primarily most of the isolates fall into what's known as clade 3. There are three distinct clades within Xanthomonas translucens, pathovirus translucens, and we want to know what's going on. Next slide. So as you can see here, these dot plots that are going around represent the tal effectors. There's 19 different tal effectors associated with the different clades. And one strain can have as much as 8 different tal effectors in these combinations. So we can see that there's a lot of genetic diversity here that'll allow it. Tal effectors are effectors that will allow the pathogen to infect the plant. So next slide. So we want to know what's the frequency of these in the field, and we have 4 fields here that we did deep surveying on, where we would go across the field and make sampling every 10 or 15 feet. And what we're finding is that the different clades are representative mostly in all of the fields in North Dakota, with some fields having, clade 3 or clade 2 and not clade 1, and only one field having really clade 1 in it. But we have a diversity of the pathogen within the fields in North Dakota. Next slide. So then we went through, we collected, isolates, B1FA, CIX 95, which comes from Minnesota. That's the one that they've been working on for a while in University of Minnesota with Brian Stephenson and Rebecca Kerlin. And then clade 1, CR22001, which is one we collected. And we ran a panel of barley to check for pathogenicity, and although they will follow a trend of virulence, the different clades seem to be behaving differently, giving different levels of virulence on each of the barley lines that we tested. GGID 01016 was one that Brian Stephenson has identified as a resistant line, and it seemed to be one of the more resistant ones between clades. But these clade differences kind of concern us in moving forward for doing screening for BLS resistance. Next slide. So our next tact is we have been transforming each of the 105 isolates that we have with barcoding and fluorescence, so that we can track how they behave in mixtures and combinations in the barley leaf. What you're seeing here is a barley leaf. The mid vein is going up the center. It's known that XTT will infect and take up the vasculature of the barley leaf and spread through the vasculature and then kind of bleed out. The green is, you're seeing the fluorescence of our tagged isolate starting to cause necrosis. Next slide. And another tack that we're taking is looking at Agilops tosii and some other wheat progenitors for non-host resistance, because XTT can't infect wheat, for resistance in wheat, where we can narrow that down. And specifically for Agilops tosii, we actually found a single line that had consistently exhibited water soaking, which means that non-host resistance actually broke down. And then we will move forward to try to identify what that loci and gene component are, for hopefully integrating that into barley either through transgenics or other means. Next slide. So with our background now of XTT isolates and the tagging platform that we're using with GFP and barcoding, we are looking into assessing the differences in the interactions between the XTT clades, how they interact in the field on a single plant, how they interact with XTU, and how they also interact with barley-associated microbes. We're using the system to track seed-to-seed transmission. So the life cycle of XTT is kind of unknown in terms of we can find it on the seed. How does it then transmit through to the next year's crop? And then we're also identifying non-host resistant genes from wheat progenitors for integration into barley. Thank you so much, Ashley, for all of this. Thank you for the BPI. Also, we used SBARE funds and the North Dakota Barley Council to help support this research.
Yeah, do you want to just mention also your upcoming research meeting?
Yes.
Yeah,
that's right. So the BLS Research Initiative will be held in Minnesota on April 5th, 4th. I have to look it up. I can't remember. It's on my calendar.
It's out on the AMBA website too, if you.
Thank you. Yeah, thanks for putting that up. And it's welcome anyone who would like to come and talk to us. Also, I think we are having it hybridized, so you can join online.
Awesome.
It's April 9 to 11.
9th to 11th. Thank you.
All right, I stopped my share, Tanner, so you can go ahead and get your slides queued up if you'd like. And while we're waiting for that, if anybody has questions for Tom, we've got a couple minutes. All right. And if there's nothing, we'll have time at the end too, just to have kind of an open discussion with all of our presenters. Oh, Nicholas, sorry, didn't see your hand up.
Yeah, no, literally up. I was just gonna ask, do we know like what the spread is? Because I don't think we've seen this in the East, and like it's possible that we've gotten it at times, and I just don't know. Like, do we know much about like where this is even? I mean, not just where it's a problem, but where it even exists.
Yeah, it exists. Yes, and we know from looking in our FHB microbiome trials that it's persistent kind of all over. We found it in New York, definitely Minnesota and North Dakota and in Idaho. The symptoms that we see, they usually follow after some kind of damage. So if you had hail damage, that's when you're going to see it the most. Late summer hail damage when that comes in, and then there's a lot of excess moisture and some humidity, then you'll see it the strongest at those points. I saw it in 20 and caused a ton of damage. And we did some pretty interesting work where we would take apart grasshopper parts, take the grasshopper parts apart and test them for XTT, and they would carry them in basically short distances, but not over long distances.
So, I mean, is part of the reason I'm not seeing it is because it's a little later in the season, and so for like the winter barleys, they're just kind of done before it even is a problem.
Yeah, that's probably a good reason why you're seeing it like that.
Nick, this is Christina. I've seen it at Plymouth.
You have?
Yeah, but very rarely.
Okay.
And the conditions were humidity and cool temperatures in the earlier part of the season.
Okay.
I think it's here, but it's not common.
Yeah, someone contacted me from USDA about it, and I was like, I don't think we see this thing. And then I spent some time looking at the symptoms, and I was like, no, I'm fairly certain I haven't seen that, especially that wet leaf symptom thing. I've never seen that. So okay, cool.
Yeah, the water soaking is very diagnostic for that. When you hold it up, and that's why it's called translucent. So you could hold it up to the light and you see that water soaking throughout. That's the tissue getting damaged as the bacterium is growing through the leaf.
Sometimes
this is Steven.
symptoms can be confused with senescence, so it's important to have like a close look into the symptoms. Otherwise, it can be confused with senescence sometimes.
Have you found any isolates, bacterial isolates from your collections, the surveys that are not XTT on barley?
When we usually find non-XTT isolates, they are either XTU or they're Curtibacterium and Pantioa.
Okay.
Tom, this is Gary Bergstrom. I've seen it on rare occasion in New York over many years, but kind of like what was recorded for North Carolina. I haven't seen it as a real issue, but occasionally see it, and you said that you found it in the microbiome, so that's consistent. Thank you.
Thanks so much, Tom. I think we'll go ahead and pivot to Tanner now. And for many of you that have been with the BPI from the start, grain genes wasn't actually originally in the original proposal. It was something that we had added in the last couple years, and finally we're able to get some funding established for that. So I thought it'd be a good time now for Tanner to come in and just talk a little bit about how they're utilizing those funds to support kind of the larger, broader Barley mission. So with that, go ahead and take it away, Tanner. Thank you.
Thank you, Ashley. So thanks for the opportunity of presenting here. So I will, grain genes talk will be a bit different than the previous talk. So I'll just give a very quick overview. Quick is the right term. So we serve many communities. Wheat is definitely is very important to us because it provides, of course, bread, wheat, and food to everybody, including kids. But we love barley. You cannot live on bread alone, and we also need to think about the needs of older kids. So we like barley, and yeah. With that, I'll just go to the grain genes. The grain genes is available, has been around since 1992. So that's like 40 years ago now. We have a website, many of you know us, and we provide information, genetic and genomic information through this centralized location. We also provide a digital platform. We have job boards, we have grains email list. We provide also spaces such as to genotyping labs if needed for them to share data. Our users are global. Most of them, we have a large population in the United States working using our tools, but other researchers around the world are also using our tools and resources. And I think this is good because more barley research will help improve US varieties as well. This is a common question, grain genes versus T3. Just want to put that slide up. We are both USDA ARS databases, but our missions are complementary. So T3 works with the raw genotypic and phenotypic data, and especially with the projects such as WheatCap, Uniform Regional Scab Nurseries, and we work with the peer review process data. So, and we have decades of genetics and genomics data. We work very closely, and there's rich cross-linking between databases. And we share resources such as genome browsers, T3 creates tracks, and we provide links so that the users can enter one database, go to another, and without even noticing sometimes that they are at different databases. Genome browsers is one area we are strong at. Most recently, we made available Barley Pangenome V2 that includes 76 genome browsers. One thing I want to mention is the AlphaFold. And so for example, Barley Morex V3, and this may be of interest to Bob when he's working on RPG7. So you can look at the gene models, and then from the gene models, you can reach the protein structure. So for example, the question will be, where is that S to P amino acid substitution will be on the protein structure. So this is also something that we can help you if you let us know, hey, this is the protein I'm working on, and can I get a predicted protein structure? So we can provide that kind of help as well. That brings me to importance of curation, because it's not just that we take the data sets and put it up. We need to create links between different data sets that may go from genomic data sets to genetic data sets. And curation is very important in that process. And this requires usually PhD level domain experts. That person needs to go to literature, understand the literature, and bring the data back. So we are grateful to AMBA for supporting halftime curator. Dr. Victoria Blake is on call, and I won't do justice here, but she worked on bringing tons of data sets for QTL, meta-QTL, both wheat and barley to grain genes. And this includes for different traits, biotic stresses, etc. Most recently, we brought this 256 meta-QTL data. This data was developed by Brian Stephenson, Gary Mulber, Ahmed Salam, and then we created genetic maps for it. So Vic also was involved creating these tracks, essentially creating links between genetic and genomic data. So you can go to this track, and then you can come to the QTL report to get more information about this QTL, and then you can go back to the gene models as well. So essentially jumping from genomic data to genetic data to genomic data again. So we asked ourselves, can large language models and AI be used as a curator or as a curator assistant? So we had Vic creating this curated data set, so we know exactly what was inside. And we applied the large language models with a conversational piece to see if we can actually extract data from PDBs as well as different tables. And this included genetic data like QTL or DNA regions or yield. We asked, for example, just based on the abstract, can we say something like, is it agronomic, biotic, and our large language model was very successful in that. And this paper, by the way, is in press and will be available soon. We also look at the different traits and then see if we can actually create data formats. And we try to hallucinate a large language model. And we were able to do it by asking about peduncle length, but in some cases, peduncle length, GPT-4 confused with panicle length, which is different. But overall, it was very successful. And we learned along the way that the way you ask questions is very important. So for example, in this case, there are instructions in orange, and then there is this range, underlying range. So we try to remove that and then add that and see how the accuracy, the correct number of correct answer will change. And once we provide the instructions, we were able to get much better data. Just compare the blue versus orange in the right hand side with GPT-4. So number of correct answers just skyrocketed, but still not 100%. So this brings up the fact that prompt engineering is very important. The way you ask questions is very important. And in coming years, we will be able to use more of these resources. And we need to ask the right questions to be able to get the right answers, especially for biological data. So conclusion, yes, definitely GPT-4 was very successful over GPT 3.5. I'm sure the next generation of models will be much better. I'll go fast. Definitely, we have global challenges. We have national challenges as well, most recently. So we are actually a data management place, and we work with machine learning, and we have the expertise, and we continue looking for collaborations, new projects, and funding to develop novel AI tools and workflows to serve our stakeholders. So with that, we appreciate data generators, curators, liaison committee, and especially AMBA. Thank you.
Excellent. Thank you very much, Tanner. And thanks for being quick with those 17 slides. You did very well.
I'm sorry, I didn't see this 5 slides rule. But
it's okay.
yes.
It's okay. No, everyone did wonderful today. So questions for him while I go ahead and I'm going to pull my slides back up, but just feel free to go ahead and go off mute and ask Tanner some questions. And there might have been one that dropped in the chat there, Tanner, if you want to look at that.
Yeah, definitely, Bob. So, yeah, Bob is saying that he's been using GrainGene's browser on a daily basis and used the AlphaFold link yesterday when we received the data showing our NL mutations. So that's very timely. Please reach out to us because when there's a mutation, then the protein structure will change. And especially with the, you mentioned serine to proline. Proline is a very rigid amino acid that breaks things essentially, the secondary structure especially. So we can just run the process for you and then create a new protein predicted structure when the mutation happens.
And I did want to just note too, I really appreciate you throwing out there just that not even a comparison, but the relationship maybe with T3, because there's been a lot of conversations internally about how the barley community would really like to embrace T3 more and make that more efficient, and how that they're definitely separate and different from grain genes, but that there's a lot of back and forth relationship there, and that we're having a lot of internal conversations with everyone involved to make sure both of those platforms are well used with barley and that we can maximize their potential. So appreciate you putting that up there as well. And definitely, if anybody else has another question for Tanner, I did put another poll up and would love for you guys to go ahead and start working on this one, but definitely can entertain another question for Tanner if you want. This is one of the polls that I've done every year, and I always like to see how this changes year to year. It has been a little different, so I'm going to go ahead and throw this out there. What do you perceive to be the greatest pest, insect, or disease to barley? All right, anything else for Tanner?
I just want to give you a little history. I don't know if you know this, Ashley, but T3 originally started with THT, which was a Hortium toolbox. From a grant we had, the Barley Cap grant several years back. It was originated here at Iowa State. So that's where it came from originally. And then it developed into bigger and bigger and bigger things, which I'm grateful for. But
yeah, I knew that it had a
barley. It started out as barley toolbox.
Yeah, I knew it had ties back to the Barley Cap, but I didn't know that it had kind of started at Iowa State. What year was that, Roger?
Oh, geez. I have to think on it. I look up when the grant was. It was
okay. No worries. I just curious.
It was in the early 2010s.
Okay. Yeah.
Yeah, there was a group of us from all over the country on that grant. So
awesome.
Minnesota, Washington, Oregon, a lot of places. 10 states, I think, at least.
Okay. Awesome. All right. Well, everyone go ahead and switch over to that poll. FHB, yeah. Even though that's the one not in the Barley Pest Initiative, still reigns probably superior as being one of the most significant pests that we have to deal with. Give it just a couple more seconds here for people to respond. These word clouds have been fun year to year just to see how they change. Different outbreaks that impact or different funding opportunities that allow you to work on these. All right, awesome. Thanks for responding to that. So FHB, reign superior. And then Barley stripe rust. Looks like it's going to be the second. BYDV is jumping in there too. All right, yep. Okay, thank you guys. So I'm going to show you now how we did spend the money. And if you remember with FY24 funds, we had $3.5 million in the distribution. This is our pie chart of our pests. And again, this is all straight from your questionnaire responses. So I always throw this disclaimer out there. I know that there's additional funds that come from other sources that go towards these pests. So I don't claim that, you know, all the dollars, all the research dollars, this is all that's going to leave for us or stripe rust or net blotch. I fully understand that there's a lot of other funding sources and perhaps also funding sources that maybe displace the need for you to allocate funds through the BPI on some of these. But just for everyone's knowledge, this is how the funds were distributed in FY24. Leaf rust actually took a pretty significant increase in terms of how many funds were spent on that disease. So we had almost a 10% increase from 2023. And we had about a 5% increase in net blotch from 2023. And ironically, even though I know a lot of you are working on bacterial leaf streak, that actually saw about a 10% decrease from 2023. So we did see some movement in terms of how those funds were being distributed. And then for this slice of the pie that's other at 12%, I wanted to go ahead and list those out: nematodes, scald, green bug, support towards the grain genes program, wet wheat stem sawfly, spot blotch, and crown rot. So that's how the 12% remaining funds that weren't in that pie chart there, those were the additional pests, which then meant kind of our orphans, or the ones that didn't receive any funding, were the hedgehog grain aphid, the mealybug and wireworm. So they were all insects this last year. But that's kind of how the distribution occurred in the funds. So maybe just if anybody has any thoughts on that distribution, any surprises there, concerns there. Just give it a little bit of time if anybody has anything they want to say about that. I will also mention, just as you're thinking, it was interesting this year, more so than years past. I've seen many of your programs diversify and maybe work on 4 or 5 pests and probably spread the funds pretty thin across that for whatever reason in this reporting year. And I didn't run the stats on this, but this is just anecdotal. Very much saw many of your programs taking the BPI funds and being more specific and targeted and working maybe just on 1 pest or just splitting your funds across 2 instead of doing 4 or 5, which obviously no problem there and how you're spending them. But I did think that was an interesting trend. And I don't know if that's what also led to some of the significant changes in the funding allocations this year. But any thoughts?
Actually, I'm assuming the 10% decrease is like a drop from 5% to 4.2%, not like 14% to 4%.
Correct.
Yeah.
Yeah. Yes. Yeah. All right. Well, if anybody has any thoughts, want to share, let me know. But then this will be, I believe, our last poll. This is really just thinking about, do you feel our work is accurately reflecting the need? So what I've done is year to year, I've looked at how we've spent the funds and then use this poll to just kind of look back and see if that's how you believe we should be spending the funds. So this right here is our work accurately reflecting the need. Here you can just drag and drop the pests. And these are the ones that are currently receiving about 80% of the funding, and just put those in rank of importance. We'll just give it a minute or 2 here for folks to do that. And I know that everyone has very diverse perspectives because you have different pests impacting your work where you're at. But I do think that this still provides some interesting insight into what you're seeing in the field. So do appreciate those of you that are willing to respond to participate in the poll.
Hey, Ashley.
Yeah.
I just wanted to say, I did not know a whole lot about cereal yellow dwarf or barley yellow dwarf. Was very recently informed that they are extremely different things.
Yeah.
And I think sometimes they get lumped together, which is interesting. And I was wondering if we wanted to add barley yellow dwarf to that, because I think that's the bigger issue we have in the Eastern US. I mean, I know we're a small market, but
I love that you brought that up, though, because there's actually been some pretty robust discussion via email about, because actually some people argued that it was one and the same, but I think there's several that believe they are different. Christina, you came off mute and turned your camera on. Do you have anything to provide on this?
My understanding, Nick, and this is, particularly from wheat, but it's the same aphids vectoring the same serotypes of the virus. So oftentimes people refer to barley yellow dwarf virus, which could be caused by any one of several serotypes of barley yellow dwarf of cereals, which could be caused by any one of several different serotypes of the virus, right? And can be vectored by any of several aphids. Cereal yellow dwarf virus can include all of those serotypes as well. And I think that the terms effectively are used interchangeably.
yeah, so I thought the same thing. And then we have a virologist here.
But two different, I think, they're not related.
Yeah.
Belong to 2 different genera in plant.
Yeah, they're not even closely related at all. They're really different, apparently. Yeah.
Kind of disease, they're transmitted by aphid species, but they are not related. They cause the same kind of disease. They are indistinguishable under field conditions.
Okay, that is not what I have seen, but
So
I'm willing to
and then-
believe that there are differences of opinion.
Yeah, well, it's interesting because these are both the perspectives that I've been explained, and that like CYDV is an overarching, BYDV kind of falls in it, and then I've had the other where they're completely separate. I actually would love to have a discussion about this with folks. I think for reporting purposes, we keep just using CYDV/BYDV as if they're one and the same, and so for just like general understanding today, when we're reporting I'm pulling CYDVB and BYDV. Sorry, I'm getting the acronyms all screwed up now, but I'm pulling them together as they're one and the same. Not that I'm suggesting they are, because I clearly cannot make that judgment call, but that's how we're reporting, just so for like purposes of today, they're included. But I love that this conversation's been happening because we've had very similar questions about this. And I think it would be interesting to pull some people together to just provide a little more clarity on this. So
Nebraska, we found more of BYDV strains compared to CYDV strains.
Okay. Great. Awesome. This is exactly the kind of conversations we need to be having. Thank you. And thanks for everyone that responded to the poll. Just looks like everyone's done answering those. We got stripe rust at the top, then CYDV, which we might also have including BYDV, bacterial leaf streak and stem rust, another rust, leaf rust, net blotch, and powdery mildew. Roger, did you have something?
You realize this is a totally biased poll.
I know, and you realize every year you say that?
I know, because it's just basically people advocating for their own disease.
And that's okay. That's okay.
I mean, you can't help it.
Yeah,
So
that's fine.
Are
you defending powdery mildew there at number 7?
Forward. It's research is a long-term mission, and
yep.
It works out different ways. You find out things you would never have known you'd find out 5 years later.
Oh, I totally agree. I totally agree. Really, the purposes of this poll is if there was a disease that was number 8 in the way we funded it, and all of a sudden it's number 1 because of importance, that tells me something. It tells me that perhaps we might need to, encourage additional investment with some of the other pests, but maybe not. So this is not overly scientific. This is really just meant for engagement and just to get interest from everyone. So I promise. I don't tell you how to spend your money, so you can do that however you want. All right,
Can I ask a question? So
yeah,
Andrew Friskcrop does this really good job of working with diverse stakeholders across the university extension teams in the US for wheat to determine losses. They don't take into account how current breeding practices has helped to avoid certain diseases or agronomic practice or like fungicide treatment. So I was wondering, is there any scope in the long run to fund a program like that for barley, where we're actually alluding to Roger's concern, that we're actually collecting that information and being able to know as certain diseases are coming up, and that we've got like a publication attached to it or some kind of DOI that's following over the years?
So Andrew is on, and I've actually asked him about this, not necessarily to do the work, but whether or not the work existed already about the economic impact of some of these diseases, because I would really like to be able to articulate that when we're speaking with members of Congress. I know that the SCAB group has done a great job being able to articulate the economic impact of these, but we have not seen that done in barley with these. And I don't know if Andrew, if you are on, if you have anything to contribute to that conversation.
Yeah, Ashley, can you hear me?
Yeah.
Thank you, Matt, for that segue, I guess. Kind of builds on what Roger was saying too. I think one thing we struggle historically in the last probably 20, 30 years is what is a justification of an economic loss? So many factors. Probably the truest way is when you get real live survey data, field survey data, but that's getting more and more difficult to obtain that due to landowner laws and how they can change amongst states as well. You can run the risk of putting yourself in more hot water than the reward, in this case, information that you're trying to gain. In North Dakota, just because our landowner laws are a little bit more lax, it's not posted unless otherwise noted. we have been able to run an IPM survey. So I can say that when I've talked to Ashley, what she's referring to, there's since 2011, I have disease prevalence data in barley. And the biggest thing is when you take severity incidence and then calculating a loss on that, it's still just kind of a conservative estimate at that point. So I mean that's available. The numbers are there. The bigger thing is someone putting in enough maybe confidence in the economic loss on that, which it's open for interpretation, but it's better than just throwing darts at a dartboard. The other thing I would say is when you look at the role of breeding and the impacts of what has resistant varieties, the other thing that we need to think about is the adoption of those varieties. Because right now we're starting to see a trend where some of our more susceptible varieties and small grains are starting to gain more acreage again. It's not just barley, but you can expand that amongst other groups too. And there's different dynamics, public, private, switch over on that. But long story short, or short story long here, more than willing to help with trying to justify and provide some of those metrics for this effort, along with the other advocacy efforts. I just need to have some help. And the biggest thing is what is achievable from what I can produce in North Dakota, but how could that relate to other barley producing regions? Okay, I'll be quiet here.
Thank you so much, Andrew. And I personally would love to see this work happen. I know that everyone's spread thin and we don't even have the BPI fully funded, but I will just put this out here now. If this is something that is of interest to you and you would be willing to help work on this, I am more than happy to facilitate a small group of folks to try to make this happen. Whether we need to pursue additional funding sources or just need some help organizing efforts and reaching out to perhaps some of the state grower groups that might be able to help us with some of the survey work, I would love to do this. But like Andrew, I certainly don't have the expertise to do that on my own, so I would really need a group to do that. So, you know, even just today, if this is something that you have interest in or think you could contribute to, definitely reach out to me because I would love to help support that. And then we can maybe do a different slide next year and Roger won't be able to complain about it. All right, we'll keep moving along here. So I did want to just talk a little bit about what's next in terms of funding opportunities. And I know that I presented this last year and been talking a lot about the Resilient Barley Initiative. But I did want to just mention, and this is more from an organizational kind of framework perspective, the RBI, just like the BPI, would receive funding through the annual appropriations budget through Congress. So we know nothing about what funding opportunities there are in FY25, other than although the Senate did not put any additional funds in the BPI budget, they actually did put $2 million in for the Resilient Barley Initiative. I still think it's going to be very hard to realize that $2 million just because of the funding climate right now. However, I'm very excited that the Senate thought that this was important enough to include. And you can see here that this is to support breeding programs, management trials, and then also to eventually have perhaps a grant program similar to the SCAP initiative. Obviously, we're not that big, but something akin to that. But the piece that I really wanted to highlight here was the $500,000 support for coordination. If we were to get any funds from Congress for the Resilient Barley Initiative, the plan is to take whatever initial funds we have and actually hire an individual to help coordinate all of that work. And I mentioned this today because the thought would be that that person would also help coordinate this work that's within the Barley Pest Initiative. I am finding it very hard to keep up with all of the amazing work you guys are doing and also being able to properly report it out and also help do some of these kind of side projects like thinking about the economic impact. Many of you are familiar with the U.S. Wheat and Barley SCAP initiative, there is a whole framework, there's staff, there's this robust structure around it. The BPI does not have that. It has literally nothing. Any support the BPI has is just what I'm trying to do in the background. And that's not really a part of my job. I'm just doing it because I think it's really important that these funds are spent appropriately and also that we keep adding to that bank. And so I want to make sure that we're articulating to Congress the good work that's happening. And so if we do get funds through the Resilient Barley Initiative, I do plan, we collectively plan to have those funds allocated towards a person that will help both with the RBI and the BPI. Now, I don't want to create a big bureaucracy around both initiatives, and I certainly don't want to suggest that there's going to be this heavy oversight over how these funds are spent. I still envision, just as you do now, you get your BPI distribution and you spend it how you best see fit. It's just going to have someone that's going to be able to help do some of this reporting, the work that I wish I could do in a much more robust fashion, but just don't have the time to. So I did just want to mention that that's tentatively in the Senate version of the ag spending bill. I don't know if it'll get through, but at least it was an indication that this is something that's important to Congress, and hopefully we'll see that to the end. And then for those of you that maybe aren't as familiar with the Resiliency Initiative, it's really focusing on those abiotic stressors, whereas the BPI is really focusing more on those biotic stressors. And a lot of this is related to climate change, and so focusing on drought and heat stress tolerance, also looking at things like pre-harvest sprout, which has become a huge quality concern in the malting industry at least. And then also looking at that winter hardiness or cold temperature tolerance piece, because we do believe that with a higher emphasis on sustainability within the supply chain, looking at winter varieties provides a lot of environmental benefit. And so hopefully bolstering some of those breeding programs that are looking at winter barley. And that's just kind of how some of the work is divvied out. And then the last thing that I wanted to talk about today, and I'm just going to do this very briefly because I do want to provide maybe a minute at the end here, if there's any closing comments from anyone else. But a few folks within the barley community have been talking about how we really want to invest in especially our early career researchers or people that are new in the industry, or perhaps even beginning farmers that are growing barley. Because right now our industry or our crop, barley, doesn't really have a program that helps bolster and support that leadership growth potential. And so this is just a concept. It's not funded yet, and we're not really sure how this is going to roll out. And so it won't probably happen for another year or two. But this is just an idea that we are toying around with and would love some feedback and contributions from you all if this is something of interest. But like I said, this would be targeting our researchers, folks that work in industry, and our farmers, and just really helping build their leadership potential. And so that not only do people want to work in barley, but hopefully we can also retain our really good talent that we're getting, because oftentimes we're losing them to other crops that have lots more money and shiny things. And so the leadership program actually would be broken down into 3 different modules. And I'll send actually this flyer out when I send a recap of the forum. And so you can look at this in a little bit more detail. But I did just want to drop this out here as I had a captive audience of barley enthusiasts, and really hoping this is something that we can get started, because I do think that we've got some great young folks working in barley, and I don't want to lose them. So a program like this might help invest in keeping them and retaining them in the barley industry. So with that, sorry, I kind of sped through there at the end. I just wanted to say, you know, many of you are currently getting funded through the BPI, but I know that you are also having to solicit a lot of other funds to support your programs. And I just want to put out a reminder. I know I sent an email to many of you that get funding through AMBA already, but for those of you that are not on that mailing, just know that AMBA and NBIC is more than happy to write letters of support. And there's quite a few NIFA grant opportunities out there right now that might be, even though I know they are a huge lift, but they might be a bit easier to get right now than additional funding from Congress. And I'm just going to be really honest and transparent about that, because I think things are going to be a little tough there. So if you do want to pursue a NIFA grant opportunity, and like I said, I think there's 3 different ones right now barley could be really attractive for, just please reach out to us. I have experience writing USDA grants, and so I understand, again, the complexities and how much time is devoted to getting those off the ground, but we would really encourage collaborative proposals coming from folks within barley. And if you need letters of support, both AMBA and NBIC is here to write those and get those in your package. So hopefully you'll be competitive. So I am just going to end there, right? Just a minute long, but I certainly will stay on for another 10, 15 minutes if anybody wants to, just for some open discussion. But I do appreciate everyone's attention today and appreciate again everyone that made presentations. It was really good to hear updates from some of the programs. So with that, I will say goodbye if you need to drop off. But for those of you that might want to stay on or have questions, feel free to just open it up.
Idea.